Hello everyone. Let me explain you about the transportation of mRNA from nucleus to cytoplasm. So in my previous video I explained you how specifically rRNA and tRNA and other proteins are transported across the nuclear membrane. Maybe it is from cytoplasm to nucleus or from nucleus to cytoplasm. In this video class, I will specifically talk about the mRNA, the transportation of mRNA from nucleus to cytoplasm. This picture tells you a story. So this big circle is a eukaryotic cell and this is a nucleus. The blue shade is the nucleus. There is a double-stranded DNA, genomic DNA. DNA undergo transcription to produce pre-mRNA or native mRNA. So this undergo processing in case of the eukaryotes to form the mature mRNA. Once after the maturation of the mRNA, it is transported to the cytoplasm to undergo translation. So I am going to talk about the process of transportation of mRNA from this compartment to this. So I have already explained you in detail in the previous video, but I'll give you an overview. This is the nuclear membrane. This membrane present around the nucleus has some channel-like structures, and these structures are called as nuclear pore complexes. You can see the whole complex is called as nuclear pore complex, and nuclear pore complex is made up of smaller units or proteins called as nucleophorins. These nucleophorins are of different types again. The nucleophorins that have abundant repeats of phenylalanine and glycine are called as FG nucleophorins. F means phenylalanine, G stands for glycine. FG nucleophorins means they have repeated units of phenylalanine and glycine. So these nucleophorins assist the transportation of the mRNA. And so this is the nuclear compartment and this is the cytoplasmic compartment. Now we have our mRNA in the nucleus. It has to be transported to the cytoplasm. Let's see how this process takes place. In case of the rRNA and tRNA, they are transported through a process utilizing proteins called as importins, exportins and RAN. Here in the mRNA, those proteins are not involved, but it involves a separate set of proteins, and one among them is mRNP exporter. M means messenger ribonucleoprotein. So instead of mRNA, it is mRNP, messenger ribonucleoprotein exporter. So mRNP exporter is a protein with two different subunits. It's a heterodimer, NXT1 and NXF1. NXT1 means nuclear export factor 1. NXF1 means NXF1 means nuclear export factor 1. NXT1 means nuclear export transporter 1. And the mRNA should be traveled to this compartment of the cell. Let's see how this works. Initially, as soon as the mature mRNA is found by this mRNP exporter, the dimeric protein, the dimeric protein interacts with the mRNA and we call this as mRNP. mRNP means, as I said, messenger ribonucleoprotein. Several of mRNP exporters associates with the mRNA. Once this association is made, we call this as mRNP. This is ready to be travel across the nuclear membrane. Now, to transport this one, the subunits of mRNP exporter protein interacts with the FG nucleophorins that are present in the nuclear pore complex. Now they interact with the nuclear pore complex and begins to move towards the cytoplasm. An important point to be noticed here is the cytoplasmic filaments near the nuclear pore complex contains a protein called as DBP5. So what we are doing is, first mRNA undergo complex formation. Once after the complex formation with this protein, the mRNA has been transported. The mRNA is shown with the red arrow indicating this. This is the cap. So mature mRNA has a cap and as well as poly A tail, as I have explained you in the previous videos. So this, call it as a mature mRNA. There is a cap and there is a poly A tail. The mature mRNA is interacting with the mRNP exporter. The mRNP exporter recognizes or interacts with the FG nucleophorins and travels into the cytoplasm. Now, when it is entering the cytoplasm, it encounters a protein called as DBP5. DBP5 is a RNA helicase. So you should know that it is a RNA helicase which runs on the RNA, that is mRNA, and removes all the proteins that are present on the RNA along with making it as a single strand RNA. You know that secondary structures can be absorbed in the mRNA or any type of RNA. Through its helicase activity, it removes all the secondary structures. Also, it removes all the proteins that are present on the mRNA. Among the proteins present, NXT1, NXF1, these two proteins are also present on the mRNA, which is assisting in the transportation, as you know. Now, DBP5 removes these proteins and delivers the mRNP. He interacts with the FG nucleophorins and travels into the cytoplasm. During the cytoplasm, it is encountering a protein called as DBP5, which removes the protein, that is mRNP exporter. After the removal, mRNA has been delivered into the cytoplasm. Now, this mRNA is recognized by ribosomes that are present in the cytoplasm, because you know that mRNA codes for a protein. The protein, that is translation process, takes place only through after the recognition by the ribosomes. Now the ribosomes are recognizing the mRNA and it is translated. But we have a nuclear protein that has transported to the cytoplasm. We should send it back. Our cells should send it back. How it does? So again, through simple diffusion process, NXT1 or NXF1 travels back into the nucleus. Once they are in the nucleus, they are ready to interact with the another mRNA to form mRNP. Through simple diffusion process, again they send it to the cytoplasmic compartment and this cycle continues. Therefore, by this mechanism, majority of the mRNAs are transported from nucleus to cytoplasm. Here, no RAN proteins are involved as in case of the rRNA and tRNA. So this completes the transportation of the mRNA across the nuclear membrane. Thank you.